everyone, and thank you for joining this webinar. My name is Zhana Kapustina. I am the Director of Product Management at Trendy Biosciences. The topic of my today's talk is addressing challenges related to high-throughput microbial sequencing. By the end of this session, you will be familiar with the semi-permeable capsule technology, how it differs from conventional water-in-oil microdroplets, and what challenges in microbiology research it will help to solve. This presentation consists of 6 parts. I will start with the overview of droplet microfluidics. I will then present an alternative compartmentalization approach, which we call semi-permeable capsules or SPCs. Next, I will overview the challenges in microbial research that can potentially be addressed once ultra-high throughput microbial sequencing technologies become more accessible. In the next 3 parts, I will overview the applicability of semi-permeable capsules to microbial sequencing and culturing, and describe Trendy Biosciences platforms for capsule generation. So let's start with the overview of droplet microfluidics, which is an integral part of what we do here at Trendy Biosciences. Many areas of modern research rely on high analysis throughput, meaning that thousands or even millions of individual assays need to be performed to collect the necessary data. Performing 1 million assays using standard tubes and pipettes is labor-intensive and very expensive, considering the price of thousands of liters of reagents. Microwell plates are a better approach, allowing the parallelization of hundreds of assays. However, working with thousands and millions of assays is still impractical. Droplet microfluidics is another step forward in liquid handling, allowing substantial miniaturization and parallelization. As thousands of droplets can be generated per second, compartmentalizing millions of assays with this technology becomes feasible. The process of droplet generation involves the dispersion of an aqueous phase, or your sample, indicated here in blue, in a continuous oil phase. In microfluidic devices, the tight control of the flow rates, as well as the appropriate channel geometry, ensures that all generated droplets are of the same size, or monodisperse. This is an important feature allowing to perform quantitative measurements, because the local concentrations of encapsulated analytes are the same in all generated droplets. Once generated, droplets serve as independent microreactors. It's important to note that the oil phase typically contains a surfactant that stabilizes the droplets, making the emulsion amenable to various manipulations. Droplet microfluidics is widely used for biological applications, and there is a plethora of published data demonstrating droplet-based assays involving cultivation of live cells, thanks to the biocompatibility of fluorinated oil and surfactant mixtures. So, why do you need to consider using droplets in your research? A major advantage is the ability to compartmentalize individual cells and either characterize them, for example by nucleic acid sequencing, or to detect a particular functional activity or surface markers. The rapid generation process and small droplet size offer a tremendous analysis throughput. Moreover, picoliter volume means improved assay sensitivity because of the increased effective concentration of encapsulated analytes, substantially reduced reagent consumption, and subsequently lower analysis cost. Trendy Biosciences offers 2 flexible platforms for all kinds of droplet-based workflows. The Onyx system is a droplet generator that is used for sample encapsulation. Onyx also supports the reagent addition applications. The Styx system is a droplet sorting platform for high-throughput screening applications. Styx detects fluorescence signals within droplets and uses the de-electrophoresis approach to actively select droplet populations of interest. Microfluidic applications supported by the Onyx system are droplet generation, merging, and pico-injection. Each of these applications requires the use of an appropriate microfluidic chip. All of these chips are available from Trendy Biosciences. The Styx system is a droplet sorter, meaning that it detects the fluorescence and deflects fluorescent droplets into a sorting channel. Here in the top panel, you can see that droplets by default flow into a wider waste channel at the top, and some of them are deflected into a narrower sorting channel at the bottom. The fluorescence is not visible under a bright field microscope, so we repeated the same experiment with fluorescent cells. Here, all droplets that contain cells are actively dragged into a sorting channel at the bottom. At the end of the run, you end up with two tubes containing your dark emulsion, or droplets that went into a waste channel, and a tube with your fluorescent positive hits that you can then analyze further by sequencing, culturing, or other appropriate techniques. An exemplary application that benefits from droplet technology is directed evolution, a method to engineer proteins with desired properties by imitating the process of natural evolution in a test tube through iterative rounds of screening. Here, library members, either protein-expressing cells or DNA containing appropriate genes, are encapsulated together with assay reagents. The emulsion then undergoes selective pressure. In other words, the assay is performed under conditions where only the proteins of interest can be functional. If the functional response can be translated into a change of the fluorescent signal, droplet sorting can be used to enrich for the variants of interest. The advantage of using droplets in directed evolution workflows is the ability to retain the genotype and phenotype connection throughout the whole workflow, even when the protein of interest is not physically bound to the cell or cell-free expression systems are used to produce the proteins. One particular example that used a Trendy Biosciences systems is the published work on the selection of T7 RNA polymerase variants with an expanded range of acceptable substrates. Briefly, cells expressing polymerase variants were encapsulated together with assay reagents. Selective pressure was ensured by the substitution of ribonucleotides for deoxynucleotides. If a particular variant was able to produce transcripts using deoxynucleotides, then a fluorescently labeled probe detected that reaction product and positive droplets were sorted. This study successfully identified mutants with reduced substrate discrimination, essentially unlocking the enzymatic synthesis of single-stranded DNA by in vitro transcription. As experts in droplet microfluidics, we love all the possibilities that this technology opens up, but we also recognize the limitations. The limitations of droplet technology derive from the fact that droplets are closed compartments, meaning that although you can add reagents inside, you cannot remove anything. Complex workflows, for example, multi-omic sequencing protocols or challenging sample types like microbial cells, are often incompatible with droplets because they need the capability of full reagent exchange. In this part of the presentation, I will focus on the properties of semi-permeable capsules. To open ultra-high throughput capabilities to new fields, such as microbiology, our scientists have developed semi-permeable capsules. Capsules are hydrogel compartments enclosed not by water-oil interface, but by a hydrogel shell. This shell is semi-permeable, meaning that large cells, particles, and nucleic acids, such as genomic DNA and mRNA, remain trapped inside, while small molecules, such as enzymes, salts, nucleotides, and primers, diffuse freely between the core of the capsule and the surrounding medium. Notably, the core of the capsule is liquid, as opposed to hydrogel beads, which are solid throughout their whole volume. The shell of the capsule acts as a very small dialysis bag and allows the multi-step treatment of entrapped samples. Capsules combine the advantages of both droplets and plates, liquid handling formats. They retain the throughput and scalability of droplets while allowing the ease of use and multi-step processing capabilities that are characteristic of sample processing in plates or test tubes. Capsules are generated using droplet microfluidics. Droplets are formed from two polymer solutions. Within a droplet, polymers spontaneously form a core shell topology, and upon brief exposure to UV light, the shell solidifies while the core remains liquid. At this point, the oil can be removed and capsules can be suspended in an appropriate aqueous buffer, for example in a reaction mixture. Aqueous buffers can be exchanged as many times as needed to complete the workflow. Upon completion, the sample can be retrieved from the capsule by mild digestion of the shell using our proprietary dissolution reagent. Capsule generation within a droplet employs the phenomenon of an aqueous two-phase system, or segregative liquid-liquid phase separation of immiscible polymers. These systems can be formed by mixing a variety of components in water, but two polymer and polymer salt system have grown rapidly. On a macro scale, the ATPS method is widely used for the separation and purification of biomolecules. The advantages over conventional extraction techniques include environment-friendly composition, low cost, continuous operation, and ease of scaling up. A major challenge related to the usability of ATPS on a micro scale is the extremely low tension of interfaces, approximately 3 orders of magnitude lower than that at typical oil-water interfaces. To stabilize ATPS-based microcapsules, photoillumination might be used to solidify the shell, given the appropriately modified polymer is used to form the outer shell. Once cross-linked, the capsule becomes stable and can be safely removed from the oil. Buffer exchange in capsules does not require any microfluidic skill. You just spin your sample down and capsules will form a pellet. You can pipette the supernatant out and then resuspend the capsules in another reagent to continue the workflow. The new reagent will instantly diffuse into the core. After cross-linking, the shell of the capsule is very stable. It withstands pipetting, thermal cycling, heating, and cooling, as well as the treatment with harsh chemicals. To demonstrate the stability of the SPC shell, we performed sample fixation in methanol and subsequent rehydration. At all stages of the workflow, capsule compartments remained intact. Similarly, 1-hour incubations at pH levels ranging from 2 to 13 did not cause any significant loss of SPC integrity. Given the discussed properties of semipermeable capsules, the technology may be very useful for microbiology. In this part of the talk, I will discuss the unsolved challenges in microbial research that would benefit from the availability of high-throughput single-cell genomic analysis methods. Microbial genomics holds the potential to address pressing research questions in the fields of microbial dark matter investigation, analysis of virulence factors and pathogenicity, microbial community dynamics, and gene regulation. Up to 99% of the microbial diversity on Earth is not easily culturable and thus constitutes so-called microbial dark matter. In recent years, cultivation-independent methods such as metagenomics and low-throughput single-amplified genome sequencing overcome the bottlenecks of cultivation and show enormous potential for the characterizations of new genomes. However, given the enormous diversity and heterogeneity of microbial communities, the lack of single-cell resolution at a large enough scale limits the interpretation of obtainable data. An accurate understanding of the evolution, acquisition, and transmission of virulence factors and antimicrobial resistance genes would greatly benefit infectious disease research. In recent years, the historical postulate one microbe, one disease has been replaced with the concept of pathobiome, highlighting that the pathogenicity of microorganisms results from highly specific interactions they have with their microbial environment and host. Elucidating these complex relationships suffers from technological barriers that do not allow accurate descriptions of pathobiome diversity. Similar limitations affect the broader research of microbial community dynamics. It is well known that bacteria belonging to an isogenic population, even under uniform environmental conditions, display phenotypic diversity and variability in gene expression. Only high-throughput single-cell techniques can provide adequate insight into the mechanisms of phenotypic heterogeneity. The technical roadblocks characteristic of conventional metagenomic and single-amplified genome sequencing techniques involved limited access to nucleic acids, bulk readout in metagenomics, low genome recovery, and low throughput. This leads to inaccurate community representation, complicated data analysis and interpretation, and simply limited informativeness. An ideal technological solution would be a single cell level readout at scale, compatible with harsh lysis conditions to improve the accessibility of nucleic acids. Single cell sequencing is not a completely new concept in microbial research, with several low and high throughput methods published to date. A relatively well-established approach is the processing of individual cells sorted into plate wells. Cells are typically stained with a non-specific fluorescent dye, such as cyber green, but they can also be specifically labeled. Fluorescence-activated cell sorting is the most common choice for physical isolation of a single cell into multi-well plates. Once isolated, cells are lysed to release the DNA. Whole genome amplification is required to generate sufficient amounts of DNA for library preparation, since atypical prokaryotic cells only contain a few femtograms of DNA. Once DNA libraries are prepared, short or long-read sequencing platforms can be employed for sequencing. This method allows the harsh cell lysis and multi-step processing of nucleic acids. However, its throughput is limited typically to 384 cells per experiment. Droplet microfluidics offers scalability and cost efficiency, which are great features for high-throughput characterization of microbial diversity. On the flip side, droplets are closed compartments, limiting opportunities to develop efficient multi-step workflows. Although reagent addition into droplets is technically possible by using special microfluidic chips, optimizations are often needed to ensure the absence of inhibitory components and the compatibility of all reaction components. Moreover, performing more than one reagent addition, with droplets becoming bigger with each consecutive step, is technically challenging. In the published example of high-throughput barcoding of microbial transcriptomes, the protocol includes 3 reagent addition steps by means of pairwise droplet merging. Features of semi-permeable capsules enable to overcome the technical challenges of both plate-based and droplet-based approaches and bring the best platform for high-throughput microbiome characterization at a single cell level. Selective permeability of the shell allows to perform each workflow step at optimal conditions and use harsh lysis to access and purify nucleic acids. All these steps are being performed without the loss of single-cell compartmentalization. Applications of semi-permeable capsules can be broadly divided into functional analysis and nucleic acid analysis. Functional screening in SPCs is possible given that the reporter does not leak from the compartment. One possibility is the expansion of microcolonies and either staining the cells or expressing the reporter inside the cell. Screening without colony expansion is also possible if there are ways of generating a strong enough signal from individual cells. Cytometry and sorting of SPCs are feasible by both fluorescence-activated cell sorting and the electrophoresis-based droplet sorting, given that SPCs are re-emulsified. Nucleic acid analysis applications depicted on the right-hand side of the slide are currently a priority for Atrendi Biosciences scientists, given their tremendous potential to address many unsolved challenges in microbial research. In the following part of the presentation, I will focus on microbial DNA sequencing using SPCs as cell compartments. Whole genome sequencing of individual microbes may be motivated by the opportunities to identify uncultured microorganisms at the species or even strain levels. Cell-specific genomic information may improve our understanding of bacterial evolution and provide accurate data on any additional DNA present in the cell, for example, plasmids and phages. High-throughput genome sequencing approach is a great tool for studying the heterogeneity of complex microbial populations. To enable these exciting discoveries and to accelerate our understanding of complex communities, Atrendi Biosciences has developed a kit for single microbe barcoding based on the semi-permeable capsule technology. First, microbial cells are encapsulated into SPCs by using our SPC generation kits and droplet generation platforms. Next, entrapped cells are lysed. Lysis buffer is then washed away and genomic DNA is amplified by multiple displacement amplification at optimal conditions. Single amplified genomes are then labeled by combinatorial barcoding, and afterward, SPCs can be safely dissolved to recover barcoded DNA sequencing. We employ the permeability of capsules to short DNA oligonucleotides to label cells by combinatorial barcoding. This method involves several iterations of randomly splitting samples across the wells of a barcoding plate, ligating barcodes, and then pulling the sample back together. As a result of this process, DNA fragments within capsules gain unique combinations of barcode sequences that allow to distinguish the genetic material of individual cells in sequencing data. To test the performance of our kit, we mixed SPCs with encapsulated Escherichia coli and Bacillus subtilis cells. The mixture of SPCs underwent the barcoding workflow, and the resulting library was sequenced on the Illumina platform. An important parameter ensuring single-cell resolution is cross-contamination between SPCs. If any genomic DNA molecules were able to diffuse and contaminate other compartments, we would see a substantial number of mixed genomes associated with the same cellular barcode. Otherwise, the majority of cellular barcodes would be associated with reads aligning either to E. coli or Bacillus subtilis reference genomes and located on the axis of the barnyard plot. This was the case for us. We observed excellent genome retention, indicating that the permeability of SPCs is not adversely affected throughout the workflow. Another important metric to assess the performance of the workflow is genome recovery. The best case scenario here is the completely random distribution of reads that would allow a particular maximum genome recovery at a given sequencing depth. Theoretical calculations show that at 1x coverage, it is possible to recover up to 63% of the genome given the random distribution of reads. Any technical biases usually occurring at amplification steps lead to loss of library complexity and hence non-random distribution of sequencing reads, limiting the coverage breadth. Biased sample preparation requires deeper sequencing to recover whole genome information, resulting in higher sequencing costs. Biased libraries would be clearly visible at the given coverage breadth versus depth graph, as points below the theoretical coverage breadth maximum are shown here as a blue dot. The best possible theoretical coverage breadth can be calculated for different sequencing depths. Here, the blue line on the right-hand side of the slide shows this theoretical maximum, assuming the absence of any read distribution biases. Shallow sequencing of our E. coli and Bacillus subtilis library showed that an encouraging result that was close to the maximum value. Each orange and blue dot here represents an individual cell. Throughout the optimization process, we observed conditions leading to suboptimal results, such as suboptimal cell lysis and loss of library complexity. Each of these issues can be decoupled and optimized individually before integrating into the workflow, as SPCs allow for the full reagent exchange. After optimizations, we have shown that upon deeper sequencing, genome recovery of more than 95% is possible from individual bacterial cells, retaining coverage breadth near the theoretical maximum. To demonstrate the feasibility of phenotyping applications, the opportunity of expanding colonies in SPCs was explored. In the microbial world, although cultivation-free genomic analysis becomes increasingly accessible, cultivation is not going to become obsolete, as the research community still needs to harmonize the discoveries made on the genomic level with downstream mechanistic investigations. This is especially challenging for extremophiles adapted to thrive at extreme environmental conditions. High-throughput cultivation of individual microbial cells allows obtaining many isolates from complex samples. Compartmentalized growth enables ample representation of rare taxa and slow growers. Microcolonies can also be used to study cell-cell interactions and perform functional screens. Cell growth in SPCs is restricted only by the physical space inside the compartment, as nutrient and metabolite exchange with the surrounding medium is happening constantly. The physical and chemical stability of SPCs allows the cultivation at unconventional conditions, such as low or high pH values. Single-cell cultivation is also relevant for eukaryotic cells. Spheroids, or self-assembled cell aggregates derived from individual cells, are used as tissue models for drug screening, tissue engineering, and fundamental biology applications. As opposed to the conventional 2D cultures or cells grown as monolayers, spheroids better resemble natural tissues due to three-dimensional interactions with the microenvironment and in vivo-like intercellular interactions. We have shown that biocompatible SPC polymers support the growth of mammalian cells and remain stable during incubation for up to 2 weeks. Single-cell cultivation applications often include enrichment as a downstream step. Semi-permeable capsules can be sorted by means of dielectrophoresis-based droplet selection, supported by a Trendy Biosciences STIX system. First, SPCs must be re-emulsified into droplets by simple particle templated emulsification that does not require microfluidic equipment. Emulsified SPCs can then be re-injected into the sorting chip and subjected to fluorescence-activated droplet sorting. Trendy Biosciences is actively working on bringing the SPC technology to the market. In this part, I will describe the available platforms for SPC generation. Capsules can be generated on our Onyx device, and this is a good option for technology developers who want to have the control over the whole process and be able to modulate SPC size. For routine sample encapsulation, we're also developing a simple push-button device that will allow sample encapsulation without any experience in microfluidics. The Flux device will be accompanied by a menu of application kits, starting from the kit for single microbe DNA barcoding. The Flux device will have the capacity to encapsulate up to 8 independent samples. Capsule size and throughput are not flexible, but the device is very easy to use. You need to push a single button and your sample will be encapsulated. After encapsulation, the device will automatically eliminate the emulsion to solidify the shell, and you will then need to transfer the emulsion into a tube, remove the oil, and suspend the capsules in an appropriate aqueous buffer. The microbe barcoding kit appeal to microbiologists working with complex unculturable communities or interested in studies of microbial evolution or community dynamics on a single cell level. The kit is designed for processing of up to 10,000 cells per experiment and yields uniquely barcoded DNA of individual microbes. This DNA can then be further processed to prepare a sequencing-ready library. A protocol for Illumina-compatible library preparation is available. Moreover, our data analysis pipeline will help you to demultiplex your sequencing data by cellular barcodes to streamline the downstream analysis steps. To learn more about our systems for droplet generation, sorting, and multi-step analysis, please visit our website at www.atrendy.com. Thank you so much for your attention. If you have any questions, you are now welcome to enter them into the chat. Thank you.